Shall we start?
Sure. Do you want me to share
No,
my
no, we will
presentation?
first introduce you, and then you can start. Yeah.
Okay. Okay, I'll wait for you to tell me when to start.
Thank you. So, good evening, everyone. We welcome you all back to our Advances in Embryology Masterclass series. So, today we have a very dynamic speaker with us, Dr. Stephen. Before Dr. Vasundara introduces our speaker for the day, I just take a couple of your time, a couple of minutes to say congratulations to Scale and to Dr. Vasundara Jagannathan because I don't think she knew this slide was there. This is mainly to say that we started the masterclass series in January and now this is the 12th masterclass. So we've been consistently having masterclasses once a month, twice a month actually, one for embryology and one for advanced ART every month. And we've successfully completed 1 year of consistency. And on behalf of Scale, I'd like to thank each one of you because we know most of you attend almost every masterclass and we know the frequent names. And I'd also like to thank Dr. Vasundara for starting this with a vision of making outstanding education available to all on a free platform. And not just Dr. Vasundara, every one of our speakers who are actually doyens in the field, including Dr. Stephen, who's actually awake late in the night for us to take this class due to the time difference. And and how are we benefiting from organizing this by spending so much time every month is only the good feeling that we're able to provide education and academic purposes for everyone. And on this note, I'd also like to give you a brief summary of what we are planning for 2026. So the master classes don't end with 2025. 2026, we have much bigger plans. We definitely have the free master classes, which we are going to have every month. And it's going to be on a different theme every month. And the first 3 months, it's going to be masterclass topics on cutting edge topics. Since we had very general topics for the first 12 months, the next year we are going to have very core specific topics. We're planning debates, we're planning dialogues, all with great esteemed speakers. We're also planning some campus courses and PG preparation courses, which are going to be hybrid and in person. So we're very excited to do this with all of you. And with no further ado, I'd like to request Dr. Vasundara to please introduce the speaker for the day.
Good evening, one and all. It gives me immense pleasure to again greet all of you for this masterclass session. December is a festival and celebration mode. Everybody is in, but still I can see all of you so enthusiastically attending the masterclass. Thank you so much, Dr. Sindhuja Srinivasan, who has been coordinating and curating the team of Scale, the technical team, the entire team. It is not a one-man show at all. And if one person has a vision, if everybody else is aligning with the vision, then it becomes quite successful. And I thank each and every one of your delegates who are spending your 1 hour, 1.5 hours of your time to train, educate, and it is going to translate into your clinical practice. And we are extremely happy, and we will continue to do this in the coming years as well with all your supports and well wishes. Dr. Stephen Fleming is well-known in the field of embryology. He's an international figure. He's a director of embryology in Cooper Surgical Private Limited. And after completing his doctorate at the University of Sheffield in 1987, he took postdoctoral research in reproductive biology, and he has various scientific publications. And from 1998 to 2008, he was a scientific director for Westmead Fertility Center in Sydney, and he was appointed as a senior lecturer in OBGYN and subsequently as an honorary associate in the School of Medicine Sciences at the University of Sydney. And he's a recipient of numerous research grants and an author and editor of several books, book chapters, and over 50 peer-reviewed journal articles. His research interests include cryopreservation, endometrial physiology, and endometriosis and oocyte maturation, and many others. So today's topic is on embryo transfer essentials, precision, timing, and traceability. Although embryo transfer may appear to be the final step in an IVF cycle, we all recognize that as one of the most sensitive and outcome-defining procedures, where even subtle variation in the technique and timing can significantly impact implantation and pregnancy rates. So we're truly honored to have you with us, Dr. Stephen Fleming, an international authority in embryology and a director of Cooper Surgical, to have this masterclass. And over to you. The virtual platform is all yours for the next 1 hour. Thank you so much for accepting our invitation.
Thank you, Vasundara. Okay, I'm going to share my presentation and get some of this stuff out of the way. So I'm assuming you can all see that okay. Wonderful. Okay, nice to see you all, and thank you, Vasundara, and the Sri Chakra Academy of Learning and Education in Fertility for inviting me to participate in this Advances in Embryology Masterclass series. I've been asked to talk about the essentials of embryo transfer, including its precision, timing, and traceability. These are the main topics that I shall be covering today, starting with the guidelines and recommendations for embryo transfer that have been proposed by various professional societies. We'll then consider the basic requirements to achieve a safe and effective embryo transfer, including those factors that may affect clinical outcomes. And lastly, we'll cover recommendations for training and competency in embryo transfer. So let's begin with the embryo transfer guidelines as recommended by various professional societies. The European Guide to the Quality and Safety of Tissues and Cells is especially concerned with the chain of custody of human material and the avoidance of serious adverse events. The HFEA's 9th edition of its Code of Practice comprehensively covers all aspects related to embryo transfer, from provision of informed consent through to multiple births and the welfare of the child. The American Society of Reproductive Medicine, the European Society of Human Reproduction, Fertility Society of Australia and New Zealand, and the Indian Society of Assisted Reproduction also provide their own recommendations regarding embryo transfer. The ASRM embryo transfer guidelines are based upon a systematic review of the evidence and conclude that there's good evidence to recommend removal of cervical mucus, transabdominal ultrasound guidance, the use of a soft embryo transfer catheter, placement of the catheter tip in the mid to upper area of the uterine cavity, greater than 1 centimeter from the fundus. And they also provide a template, which is particularly useful, of the 12 essential steps to embryo transfer, from the preparation for embryo transfer through to the immediate ambulation following embryo transfer. And some of these essential steps we'll be covering later in this presentation. So what should be your lab plan for your embryo transfer? Well, it's really important, of course, that you have a clear standard operating procedure describing all the steps leading up to the transfer and following the transfer. So this should include a clear written plan for each patient, discussion, of course, regarding the number and quality of the embryos to be transferred, and a decision regarding the fate of any remaining embryos, whether you should culture them on, vitrify them, or discard them, or indeed donate them for training and research. Consultation and confirmation of the embryo transfer plan with the clinical team is really important, and checking that all the necessary consent forms have been signed by both the patient and the treating clinician. And then confirmation of the embryo transfer plan with the patient. And last, but certainly not least, ensuring traceability of all the steps during an embryo transfer. Now let's consider the different features of embryos selected and some of the differences between fresh and frozen embryo transfers. So the stage of embryo for transfer could be the cleavage stage, the morula, or the blastocyst stage. And of course, the embryo might be a fresh or cryopreserved embryo. It may be not biopsied or biopsied. It may have its zona pellucida intact. It may be hatching, or it may have hatched free from its zona pellucida surrounding it. So the selection of the embryo is based upon its developmental stage, morphological aspects, cleavage patterns, and if you have time-lapse incubation, morphokinetic parameters. There was a Cochrane review by Gluchowski et al. Back in 2022, and they found that the live birth rate following a fresh embryo transfer was higher in blastocyst stage transfers, suggesting that if there was a 31% of women achieving a live birth after a cleavage stage embryo transfer, then 32 to 41% would do so after a blastocyst stage transfer. And they also found that the clinical pregnancy rate was higher in blastocyst stage transfers. And that suggested that if 39% of women achieved a clinical pregnancy after a cleavage stage transfer, then 42 to 47% would do so after a blastocyst stage transfer. However, based upon this systematic review, they were uncertain whether blastocyst stage transfer increases the cumulative clinical pregnancy rate, but interestingly, their post hoc analysis suggested that vitrification combined with multiple embryo transfers could do so. Now, comparing fresh or frozen embryo transfer in this Cochrane review by Zart et al. Back in 2021, they found little or no difference in the cumulative live birth rate between the freeze-all and conventional strategies in 8 randomized controlled trials of 4,712 women, suggesting that for a cumulative live birth rate of 58% following the conventional strategy, there would be a cumulative live birth rate of 57 to 63% in the freeze-all strategy. However, based on 6 randomized controlled trials, including 4,478 women, they found that they might develop less ovarian hyperstimulation syndrome after the freeze-all strategy. And they suggested that in that case, if there was a 3% of ovarian hyperstimulation in the fresh transfer, it might only be 1% following a frozen embryo transfer. Now, though frozen embryo transfers do reduce ovarian hyperstimulation and enable, of course, pre-implantation genetic testing for aneuploidy and fertility preservation, programmed frozen embryo transfer cycles have also been associated with an increased risk of pregnancy-related hypertensive disorders such as preeclampsia and postpartum hemorrhage, as well as various other risks such as a higher mean birth weight, an increased risk of prematurity, and of course a longer time to pregnancy. Now, since ovulation is suppressed in the presence of exogenous estradiol and progesterone in a programmed cycle, higher rates of preeclampsia may be due to the lack of a corpus luteum, which we have to remember not only provides estradiol and progesterone, but also produces vasoactive factors such as relaxin and vascular endothelial growth factor, which can combine to help with the early stages of implantation. So what about the numbers of embryos for transfer? Well, regardless of the embryo development stage, the recommendation is that labs should strive to use a single embryo transfer policy to avoid multiple pregnancies. So the decision on the number of embryos to transfer should be based on the national and local regulations. So, for example, in some countries, you're not allowed to transfer more than 2 embryos without special dispensation. The quality of the embryo, the age of the patient, the ovarian response, and the rank of treatment. But regardless of the embryo quality, number or stage, the transfer method remains the same. So what about the equipment, media, and disposable required? So you need to ensure that you have a sterile work area, that you can maintain temperature, pH, and osmolality. So really you need to be using a workstation or a biological class 1 or 2 cabinet to maintain that sterile environment. You need heated surfaces and heated microscope stage for maintenance of the temperature. And you need a stereo zoom microscope for identification of the embryo, and so you can observe the embryo entering the catheter. You want to have a warming block or an incubator for placing cap tubes containing your embryo transfer medium. And you need your incubators gassed for stability of pH and temperature. And these might be placed both within your workstation during the embryo transfer and, of course, for prolonged culture for other embryos following the transfer. Other disposables that you will require will be Petri dishes. So this is for flushing the embryo transfer catheter into. Of course, a dish for loading the embryo. So a transfer dish for loading the embryo into the transfer catheter. Also a dish for washing the catheter after loading if you're using an oil overlay. And checking the catheter after the transfer for maybe retained embryos. You'll need a 1 ml or smaller syringe with a Luer lock, and of course an embryo transfer catheter with or without an inner catheter. Personal protective equipment should be worn, including the recommendation is powder-free and latex-free gloves. I'll come on to that later, which are generally tend to be non-toxic. Of course, a waste bin and non-toxic cleaning products. So you should clean between each embryo transfer. The embryo transfer medium needs to be able to maintain physiological pH. So if you're quick, you can use bicarbonate buffered medium. Otherwise, you maybe need to be thinking about using a zwitterion type buffer system, such as HEPEs or MOPs, or combination of the two that are not dependent upon the CO2 tension to maintain physiological pH. You can also use specific transfer media with increased hyaluronan content, such as embryo glue or uterine transfer medium, because there is evidence that they appear to be beneficial. But that actually may depend upon the patient's history and etiology, so not necessarily going to be beneficial in all patients. And you have to remember that some culture and transfer media indeed do contain hyaluronan in any case, and you can see some examples of this on the right-hand side. It has in the past been suggested that there may be possible benefits of increased viscosity. So when we look at studies looking at this, to give you some idea, complete culture media such as G1+, G2+, GTL, one step, global total HEPEs, and sperm wash medium, they have a range of very closely related dynamic viscosities, which are known as centipoise. Estimates of 1.65, 1.77, 1.68, 1.29, 1.18, 1.15, 1.20. So all very, very close. Complete transfer media, such as embryo glue and UTM, have viscosity estimates of 3.59 and 1.28 respectively. And with increased serum, so for example, with Global's HEPEs medium, with 10, 20, 30, and 50% synthetic serum substitute per volume, they have viscosity estimates of 1.16, 1.23, 1.25, and 1.34 centipoise respectively. So I think you can see that these data demonstrate the vast difference between the viscosities of all these embryo transfer media and the assumed viscosity of uterine fluid, which is 1,000 centipoise. So it's a vastly different viscosity between the transfer media and the uterine fluid. In terms of the higher protein concentration, well, a study randomized 750 patients into 3 groups according to the concentration of synthetic serum substitute in embryo transfer medium, 10, 20, and 50%, and the rates of clinical pregnancy were 44.61, 48.79, and 45.49%. And live birth rates were 41.67, 43.96, and 41.7%. So, in fact, no significant differences in pregnancy or live birth rates, regardless of the concentration of serum. And in fact, back in the 90s, we used to transfer embryos in 100% serum, and I don't suspect that made much difference either. Now, interestingly, if we think about frozen embryo transfers, we have to remember that a frozen embryo transfer subjects embryos to the osmotic and oxidative stress of vitrification and warming. And this longitudinal study carried out in Japan analyzed clinical outcomes of blastocysts that had been cultured in NACA one-step medium, then vitrified if suitable, followed by recovery culture in either NACA one-step or SAGE one-step medium containing GM or granulocyte macrophage colony stimulating factor or GM-CSF. And that recovery culture was for 3 to 5 hours post-warming and prior to frozen blastocyst transfer. And they found, as you can see in this table, that the biochemical, clinical, ongoing pregnancy and live birth rates were all significantly higher following recovery culture in the media containing GM-CSF. So what's the likely explanation for a beneficial effect of GM-CSF during recovery culture of vitrified warmed blastocysts? Well, the cytokine or embryokine GM-CSF is a survival factor, which fine-tunes gene expression, protects embryos from cell stress, and promotes normal glucose uptake, which in turn enhances cell survival and facilitates on-time development, implantation, and development of a robust placenta. Now, when we come to the quality of disposables, these tables here, these come from a study by Martin Nyes et al. And this was a prospective 4-year toxicity survey of ART consumables. So this was 36 products from 72 brands, and they were assessed using sperm survival testing after 24 hours and 96 hours of exposure. And their study found that 13, so almost a third, of these products to be reprotoxic. So they defined these products as being reprotoxic if they had a sperm survival test index less than 0.85. So this is the sperm survival with exposure to the product divided by the sperm survival in the absence of the consumable. So if the performance in the presence of the consumable was less than 0.85 compared to the control, then it was considered reprotoxic. And what they found was that this included 8 brands of unpowdered surgical gloves and 1 type of embryo transfer catheter. On the other hand, pun intended, a randomized controlled trial by Hanoun et al. Of powdered versus unpowdered gloves at the time of embryo transfer in 712 participants actually found no difference in the clinical pregnancy rate. So I think we probably are waiting for a bit more data on some of these factors. So next, what are the many aspects that need to be considered in preparation for an embryo transfer? Well, as I said previously, you need confirmation of patient consent to the embryo transfer. Usually this befalls the embryologist to check this. Confirmation, of course, that you have all the equipment and disposables required. So preparation of the embryo transfer medium with or without an oil overlay in the transfer dish, off-gassing and preheating of the dishes, and of course, preparation of the embryo transfer catheter and syringe. Confirmation of embryologist identity and availability for embryo transfer, timely switching on and checking the temperature of key equipment for the embryo transfer, and very importantly, checking the theatre list to determine the order of patients for embryo transfer. So this is one way of mitigating a mix-up in terms of embryos and patients. And to properly check for this, double witnessing of the patient's ID and the patient's plasticware labeling. Effective communication is paramount to avoid ambiguity and error. So this should be part of your policy and process, your lines of communication, and custody of the patient's embryos. Now, to avoid embryologists being rushed into doing the embryo transfer, it's actually very useful to have a timeout before commencing the embryo transfer. And during this timeout, you can confirm the number and quality of embryos to be transferred. And then subsequent to that, you can make sure that you have synchronized timing or pacing of the preparation required for the embryo transfer by the clinical team, for example, cleaning of the vagina and cervix and mucus removal, and the loading of the embryo transfer catheter by the embryologist. And after the embryo transfer, then lab confirmation is required that there's no embryo retained within the catheter, and the patient should be notified that the embryo transfer has been completed and what is going to happen to their supernumerary embryos. But perhaps the most important aspect of embryo transfer is ensuring traceability of the embryo's identity. Indeed, traceability underpins all of our quality actions in the lab. It relates to all aspects of the operation of the IVF lab and clinic and covers all the clinical material, the reagents, consumables, and equipment, and personnel who come into contact with these various items, all the data that we collect, record, or even destroy, all paperwork and records, so current and archived SOPs, training competency records, equipment procurement, qualification and maintenance, non-conformities, root cause analysis and corrective actions for adverse events, and regular audits and documentation of meeting minutes. So nowadays, manual double witnessing is often supplemented or even surpassed using electronic witnessing. So electronic witnessing typically uses either a barcode-based or a radio frequency identification or RFID system for identifying that particular patient and their related plasticware. So at the start of the cycle, you'd have double witnessing to relate a given ID card, for example, to the female patient, and also the ID, for example, of the semen pot to the male patient. And from there, it's merely a matter of matching any other consumable to that initial ID so that you maintain that chain of custody. So that could be through insemination, all the key steps really, especially insemination, embryo culture, biopsy, and even cryopreservation. So when it comes to the embryo transfer, for example, we would have a patient come into theatre, they would show us their ID card, we would read on the other side of their card their name and their address, and we would confirm that with them. And then we would put it into the reader in the lab, where we would then bring out the embryo transfer dish, which was also labeled and tagged. And then we would have a match between the ID card and the embryo transfer dish. And in addition to that, we would have a camera system that the patient could see the ID on their dish and confirm to the clinician that their ID in terms of the name, date of birth matched what they were expecting. So if you have a really tight system like this, you can avoid any of those terrible instances where unfortunately, patients end up with the wrong embryos transferred. Now let's look how a typical embryo transfer might be performed. So what I'm going to show you is an illustration of how an embryo might be loaded. In this example, it's using a Wallace Shaw Pro catheter. Its sterile triple pouch supports aseptic technique, allowing the clinician to take the outer sheath into the theatre to navigate the internal loss of the cervix, while the embryologist uses the supported inner catheter to easily load the embryo for transfer. So this is showing the loading of that transfer catheter. So here the embryologist is just unpackaging the outer, or the packaging containing sterile packaged outer and inner catheter. So he's put the inner catheter in the laminar flow cabinet ready to use, and he's going to pass the outer catheter to be taken into theatre for the clinician to navigate the internal loss. And usually we would wait until the clinician tells us that they've managed to do that before we bring out the embryo in its culture dish. So once everything's been unpackaged, these packagings are not sterile, including the cap of the culture tube here. So once everything's been unpackaged, then the embryologist would bring out the culture dish and, importantly, don surgical gloves so that the process can be carried out aseptically. Now you notice that this the Braun inject syringe doesn't have a rubber plunger. So the first thing is to take up some transfer media in that syringe, tap up any bubbles that might be in the syringe, and remove those bubbles before attaching it to the inner catheter. So you can see this catheter supporting by that metal tubing, which is actually very useful when it comes to probably the hardest bit, which is loading the embryo into the catheter. So now the catheter can be flushed clear with that transfer medium to remove anything undesirable that might still be present inside the catheter. Now the embryo can be loaded into that catheter. And the method that Dave Morrell is using here is a medium air technique. So that gives you a bit of a sense of security that the catheter will retain the embryo as you take it through to theatre. Okay, let's go on to the various factors that may or may not influence the outcome of an embryo transfer. So temperature, of course, must be maintained as close as possible to 37 degrees C at all times, and pH within the certified range for that specific culture media that you're going to be using, both within the transfer dish and for the transfer. So you need to validate that both of these are maintained where they should be, for example, with temperature, using a thermocouple, and pH, of course, using what we'd recommend would be a gas monitor for that. Osmolality is a bit more difficult. I mean, that you want to maintain this close to a certified range. Osmolality is the measure of the total osmotic pressure in a solution, which is largely dependent upon the combined concentration or molarity of its organic ions. And among those for which data are available, the lower acceptable limit of commercial human embryo culture medium of lowest osmolality, so for example, G2 with HSA, is 248 milliosmoles per kilogram, while the upper acceptable limit of the highest osmolarity, Rusty Pool's P1 medium, is 298 milliosmoles per kilogram. Sterility, as I said, must be maintained. So use a laminar flow cabinet, use surgical gloves, and cleanliness must be maintained, of course, to avoid toxicity and microbial contaminations. This is where it's important to clean between cases. So what is the worldwide variation in embryo transfer method? So you can see the data here in this chart showing a web-based survey of 265 centers in 71 countries around the world. And although there were quite high percentages of these various methods for washing the catheter, etc., you can see that they weren't 100%. And indeed, a 38-question electronic survey of embryo transfer practice in 79 UK HFEA registered clinics revealed a wide range of variability and lack of standardization in embryo transfer technique, with more than 70% discordance. Ultrasound-guided embryo transfer was performed by 83% of practitioners, with 42% using a tenaculum and 22% proceeding with the embryo transfer, regardless of the presence of fluid within the endometrial cavity. Embryos were deposited into the upper middle portion of the uterine cavity, but interpretation of placement ranged from 0.5 to over 2 centimeters from the fundus, with 68% of practitioners allowing immediate ambulation following embryo transfer. So I think you can see that there's still a need for standardization of the embryo transfer procedure. The type of syringe used for embryo transfer can vary. It should, of course, be non-toxic. It may be disposable. It may be reusable. It may be glass or plastic. It may have or may not have a black rubber stopper. If it has a black rubber stopper, then you really should do a toxicity test on that rubber. The size or volume of the syringe might be 1 ml or smaller typically, and it problem. They tend to have not quite as smooth an action as those with a rubber stopper. And I'm hoping you can still hear. Yes, you can. And it should have good control. So in fact, there's been an interesting study using a mouse model for in vitro simulation of embryo transfer to investigate the impact of varying injection speeds on subsequent embryo development. In that study, pre-compaction 6-cell or greater embryos were loaded into a catheter, and they were connected to either a 1 ml insulin syringe or a micro syringe with a plunger diameter of 1 millimeter. The speed of injection using the insulin syringe being around 1 meter per second, and that of the micro syringe being less than 0.1 meter per second. And subsequent embryo development rate was in fact slowest in embryos that were exposed to the faster embryo transfer. So don't do what I once saw, which is a clinician banging the plunger on the catheter like that. You want to do a very gentle and slow injection of the embryo. In terms of the catheter, there are many options available to you: soft versus stiff catheters, 1-stage versus 2-stage. Of course, you may need to use an obturator or stylet. For difficult transfers, there are various sizes. Some are echogenic, which is particularly useful using ultrasound guidance, versus those that are non-echogenic. And then there's going to be various variations in terms of cost and packaging, etc. The clinical team preference should ideally be consistent, which helps avoid mix-ups or issues with the catheter. So it's nice if everyone can follow the same SOP, have the same training, so you can check for competency, KPIs, and internal audits and retraining. So in terms of the catheter loading, you need to select the catheter type and syringe type. And the recommendation is that you do rinse the catheter before loading the embryo. And as I said, that can help remove any substances or debris that may be present within the inside of the catheter and remove their deposition into it. And indeed, a survey demonstrates that most labs do catheter. In terms of loading of the embryo transfer catheter, you can see from this diagram on the right that there are many different approaches, either using the embryo transfer medium or embryo transfer medium with air bubbles in various positions. And in fact, there's little consensus with little evidence that any method is better than any other. And various studies, including meta-analysis, in fact, revealed no clear advantage of any of those methods for loading the catheter. And this systematic review showed again that there was no difference in terms of the live birth rate between the air embryo transfer fluid versus embryo transfer fluid only models. You can see that that there's no difference here in these charts. In terms of the transfer volume, that's kind of a bit more interesting. There are actually few randomized controlled trials or prospective studies. A survey showed that the majority use up to 300 microliters. 19%, in fact, use 300 to 500 microliters, but only 1% use 500 to 700 microliters. And there was concern that less than 10 microliters may decrease implantation rates. And we've always been concerned with large volumes and the perceived risk of expulsion of embryos beyond beyond the uterus and maybe cause a tubal ectopic pregnancy or maybe a retention in the catheter. So typically 20 to 30 microliters is used in the catheter as the transfer volume. And indeed, one study has shown that you achieve better outcomes using 40 to 50 microliters compared with 15 to 20 microliters. So this was in the implantation rate of 24.4% versus 14.7%, and pregnancy rate of 40% versus 23%. So both were significantly different. What about with loading embryos directly from microdrops from the culture dish with an oil overlay? Well, one study has compared these outcomes from this and found, in fact, no difference in clinical pregnancy rates. So if you want to use the advantage of an oil overlay, there's no reason why you shouldn't. However, if excessive oil is inadvertently deposited into the uterine cavity, there may be potential risk for a reduction in endometrial receptivity, especially for hatched embryos that are not protected by the zona. And it's possible also that the oil could absorb steroid hormones such as human chorionic gonadotrophin, which have both embryonic and maternal sources. So the part of the catheter that comes into contact with the mineral oil can be rinsed prior to the embryo transfer procedure. In terms of checking the catheter after the embryo transfer, again, in your SOP, there should be a clear description of the steps required to follow in the event that an embryo might be retained in the catheter. So, of course, you observe the tip of the catheter as you flush out the remaining media in the catheter into a Petri dish. Check particularly if there's any mucus surrounding the catheter tip and flush medium in and out of the catheter to check for the possible presence of a retained embryo. And then notify the clinical team whether or not there is a retained embryo. So the patient is waiting and the clinician is waiting for that confirmation. If there is, it's then necessary to recheck patient ID because you probably put the transfer dish back into the incubator. And you need to reload the catheter and retransfer the embryo. And you can either reuse the same catheter or a new catheter, but ASRM guidelines suggest 67% of clinics use a new catheter, and we would certainly always have used a new catheter. As to who actually injects the embryos, this sort of varies from country to country, center to center. There's no evidence either way. And as I've explained previously, gaps in volume don't seem to have much impact. Speed of injection has been shown to impact lysis rates of embryos, so that needs to be properly controlled. And I remember that tuberculin syringes have no recoil, but insulin syringes do. So in that case, you need to maintain pressure on the plunger. So the clinicians needs to maintain pressure on the plunger during withdrawal of the catheter from the uterus. So in some instances, it all depends on who's going to control this best. So let's lastly think about what's required to train and become competent and remain competent in embryo transfer. Now, it's not surprising, perhaps, that there's no internationally accepted practical training to cover lab techniques and skills with an established minimum number of processes that are considered prerequisite to acquiring a license for performing clinical embryology. And of course, this applies also to embryo transfer. So really, it tends to depend upon training in technique and acquiring knowledge from more experienced colleagues in an adequately equipped laboratory. Although there may be multiple publications on training for clinicians, there are not as many for embryologists. Now, this is just a scheme that we put together for training in a whole range of techniques, but this would include embryo transfer. So the flow that we suggest is that you start with understanding the laws and guidelines and being aware of the scientific literature, which I've sort of addressed to some extent in this webinar. You then progress to a tutorial with covering your protocols, your documents required, the preparation that's required, etc. And then you can go on to training where you typically would observe embryo transfers being carried out. And if you want to get hands-on training, then it's sometimes useful to do external trainings. You can go on an embryo transfer course. You can use Vertimed simulator training. It's particularly useful. We conduct those trainings ourselves. Or if you're fortunate that you have a good relationship with another lab, you can visit an external lab. And with your preclinical training, of course, you can use blue bees, for example, or animal embryos, or donated human material that's been donated specifically for training as part of your preclinical training. And it's important that you keep a logbook where you record all of the training that you've conducted, including the numbers, and that it's signed off by the person that's responsible for supervising that training. And that logbook would cover everything from planning all the way through to troubleshooting, etc. And then you can progress to your clinical training. And so typically this would be done under supervision, and this would be a stepwise training, and then you'd have typically annual checks of your competence using objective key performance indicators. So the key performance indicators, this is actually for frozen embryos, but the key performance indicators for frozen oocytes and embryos would be, of course, the transfer rate or the implantation rate. So when do you decide the competence has been achieved? Well, in advance, you need to define your determinant for competence. And this could include things such as, the embryo not being retained in the catheter, the time that it takes to do the loading of the embryo, of course, the implantation and clinical pregnancy and ongoing pregnancy and live birth rates. And with your KPIs and performance indicators, you should always use a reference population, which are good prognosis patients or gold standard, or you can define your own reference group, and then log your specific outcomes and the number of training cycles. So how to train an embryo transfer? Well, you need to do all of this. You need to go through from reading through and understanding the SOPs and training alongside your trainer, performing all of these different procedures, including the witnessing of all steps. And you can monitor your lab and clinical outcomes using a control or Short chart such as this. And you can plot these outcomes over time against a mean that's been derived from your previous year's results. So this could be applied to your embryo transfers as well. So you can see when things are maybe going out of your 3 standard deviations, your control limits, where you've urgently got to do something about it, or you may be pleasantly surprised that you're performing above where you were performing previously. So let's summarize what we've covered. We have to recognize that the embryo transfer is a multidisciplinary process. So it does require coordinated planning and communication between all staff members, admin staff as well. We have to remember that the embryos are very sensitive to any fluctuations in temperature, pH, and osmolality. So we need to do our best to control those and minimize the time these embryos are out of the incubator. It's also oxidative stress that we're exposing these embryos to. Equipment and consumables must be prepared well in advance. And really importantly, we must maintain that chain of custody of the patient's embryos. Of course, it's vital that we have a systematic training and checking of ongoing competency in embryo transfer. And there are several best practices that I've tried to reveal to you, though the data is limited in some areas. So I hope that was useful to you, and I will stop sharing there, and I'm happy to take any questions that you may have.
Thank you, sir. That was a very elaborate and clear presentation. So shall we take the questions on the chat box first?
Yes, I should look in the chat box, shouldn't I?
Yeah. So the first question is, what is the disadvantage of using HEPIs during during embryo transfer, besides its advantage of maintaining pH for a longer period than one-step media?
Yeah, that's a good question. The only real disadvantage of using EPs during an embryo transfer is, of course, the fact that the HEPIS buffered medium will not be exactly the same as your culture media or your transfer media if your transfer media is different to your culture media, typically in terms of its osmolality. So there may be a little bit of osmotic stress moving from one media to the other. So that's a very, pertinent question to ask. I mean, I actually came across one study where HEPIS buffered media was suggested to adversely impact fertilization rates with ICSI. So, we have to be aware of the fact that that there may be disadvantages to some media. So you need to do a risk analysis and decide, well, what's the greatest risk? Is it maybe the slight osmotic stress that you're subjecting the embryo to versus running out of the range of physiological pH? So we know that physiological pH is very important.
Yeah. So when we're at that question, so besides loading the embryos in HEPIS buffered media, some people use HEPIS buffered media for flushing out the outer and inner catheter first, and then they load the embryos with culture media. Is that okay?
I think so. I mean, for example, I think embryo glue is recommended that you flush the catheter with embryo glue, and goodness knows if you flush the catheter with stage 1 step GM-CSF, you could imagine that this becomes very expensive. So I don't have any problem with using one of the more regular or cheaper media for flushing the catheter. That you're doing this for a different purpose, and if there's any of the media retained in the catheter, I don't think that's really going to have a significant impact on the media that you load with the embryo.
That brings
Especially if
me
you're
to the
loading like
question.
20 to 50 microliters of media with the embryo. I don't think it's going to have any adverse impact. So I would have no problem whatsoever with doing that.
So that brings me to the question, do we absolutely need to flush the outer and inner catheter before we load the embryos? Because some people prefer.
Well, yeah, you don't absolutely need to, no. But it's just, one of these precautionary things that many labs do. Some labs go to the extent that they'll actually wash the culture dishes before they prepare culture dishes for embryo culture. So, it depends how far you want to go with this, but it's pretty easy to do. It's quite nice in a way that if you have flushed the catheter, I I suspect that then, when you come to load the catheter, there's going to be less problems with um, unwanted air bubbles, let's say, appearing inside the catheter, especially if you're using a more viscous medium for flushing the catheter. So, um, yeah. So, all these catheters should have been tested for toxicity, and they should be clean. But you're just you're putting all your trust in the manufacturer in that instance to if you do that. And most of us embryologists with many years experience prefer to avoid a problem than have to solve it. So we tend to flush catheters.
Especially with the
It's
embryos being
it's an easy thing
exposed.
to do, and it's one less thing to worry about.
So you mentioned in one of your slides that the dishes are generally coated for cell culture, and so it's not ideal to use the surfaces which are not coated. Can you be a bit more specific about that?
Yeah, so this applies not just to embryo transfer, but it applies to micro manipulation, whether it be ICSI or biopsy. So there has been a tendency in the past for some labs to just take a standard Petri dish, a higher one, and use the lid as a micro manipulation dish. Now, the risk that you take if you do that is that you're actually using a part of the Petri dish that has not been tested for toxicity. So when manufacturers test the dishes, they only test the part of the dish that the embryo or the oocyte is going to sit in, not the lid. So again, it's just you hope that all the components of that Petri dish, including the lid, are the same, but you can't be certain that that's the case because it hasn't been proven to be the case in testing. So the same applies to embryo transfer. So, what I would do is I would use the lid maybe for flushing out the catheter. It doesn't matter, it's just waste medium into the lid of into the lid of the dish. But anything that's important, for the embryo that the embryo comes into contact with, use the bottom of the dish itself. And the same applies to a transfer syringe. I don't know if it's one of the questions, but if you're using, for example, a transfer syringe that has a rubber bung, as long as the media doesn't come into contact with that rubber bung, you can use it confidently. But if you, for example, tip up the syringe and tap it to remove the air bubbles, the media comes into contact with the rubber bung, then you flush the catheter, then you may be unwittingly introducing a problem. So again, you can just be smart about how you use some of these consumables.
So in the chat box, there's a question. Can we use GTL media for loading and transfer of embryos?
Absolutely. So, there are a few media that are specialized for embryo transfer, like such, as I said, embryo glue, for example, and uterine transfer media, both of which contain high hyaluronan. But there's no reason why you shouldn't just use any culture media for doing a transfer. So again, back in the 90s, for example, we used to use Origio Universal media, literally universally. So we used it from everything from sperm preparation all the way through. Well, actually, I'm telling a bit of a lie because we did use 100% serum for the transfers. But if we had wished to, we could have used that for the transfers as well. So it's just a transport medium. Essentially, that's the most important function, is to transport the embryo into the uterus. Any other putative advantages of one medium over another for transfer? I would say watch this space because I've written an article on this. There's a lot of interesting science behind this, but essentially, the standard patient, good prognosis patient, they will do as well with a standard culture media as they would with anything else. So no reason why you shouldn't use that.
Vasundara ma'am, you want to take the remaining questions? There's a question about it.
So you were having a discussion about GM-CSF in the culture media, right? So that was a study which is very promising, showing very high clinical pregnancy rates, live birth rates, and everything. So do you think a brief exposure of 3 to 5 hours is going to really, improve the embryo competence? That is one. And whether we have to use it in specific group of patients with RAF and advanced maternal age. And also whether we don't know about the long-term epigenetics and obstetric outcomes and things like that. So it was a cohort study, I think. So could you show
It was. Well, all I can say for Vasundara is that study has been replicated now by a number of centers in other countries, so not just one other country. And I just recently got a report from a group in Australia that have been trying to see if they can, in their hands, replicate that Japanese study. And now we know, and you'll see them presented at meetings and hopefully eventually published. We now know that it's not a one-off, so it's very interesting. It does appear that this is a survive. What we have to remember is that when we vitrify and warm a blastocyst, we get these fantastic survival rates, apparently, but not all of them produce pregnancies, and certainly not all of them produce ongoing pregnancies. And what we can't see so well, and particularly within a blastocyst, and particularly within the inner cell mass of the blastocyst that is surrounded by the trabectoderm, is we can't see for sure whether all the cells have actually survived. And we certainly can't see whether cells are starting to undergo apoptosis or cell death. So we transfer these embryos thinking, yeah, they've survived fine. We transfer them, and then it's pretty much in the lap of the gods, whether these embryos produce a live birth or not. So it seems like GM-CSF is doing precisely what it is designed to do. It's designed to act as a survival factor, and it's designed to mitigate stress that tends to drive cells or tissues to apoptosis. So it may well be acting as it should in these blastocysts, and thereby improving the clinical outcomes. Whether it's necessary in all patients, I think, like all of these, we call this an add-on, like all of these additional treatments that we provide, they're certainly not necessary for all patients. It's more likely, for example, we know that the ability of oocytes to survive injection is not so good in older patients. It may be that the ability of embryos to survive vitrification is not so good in older patients. So, maybe that there are certain groups of patients where it's going to be more beneficial than others. And in terms of your last question, which was about unknown long-term potential impacts on epigenetics or whatever, I think the only reassurance I can offer, I suppose, is that this is a naturally occurring endogenous cytokine in the reproductive tract that embryos are normally exposed to in any case. So it's not as if we're introducing some weird and wild synthetic manufactured compound that the embryo would never normally see. So, and there have, of course, have been safety studies conducted in both the mouse model and the human, and there's no evidence of any adverse outcomes in terms of, for example, at least at a genetic level, in terms of chromosome numbers, that kind of thing. So epigenetics, it's less sure. And if you want to get into the field of epigenetics, you could pick any in vitro process you want, and I can show you data in mouse models, for example, where there's an epigenetic effect. Simply culturing embryos through to the blastocyst stage, are we selecting for embryos that are more likely to be prone to cancer in future because these cells don't die, they keep dividing. So I mean, all these things we can worry about, but as yet we just don't know, and we have no idea. It's a relatively young field, only began in 1978, so we still don't know really any long-term impacts of anything that we do in vitro. So all I can say is I don't know the answer. We don't know the answer, and all we can do is keep monitoring and hope that all is well, and in the meantime, getting the benefits of these various treatments.
So we have a lot of catheters like, Wallace, SurePro, Cokes, and so many catheters that is available. So does the loading technique anyway significantly vary across catheters because of the curvature or anything like that?
No.
Or is it the same across the catheters?
No, I mean, the catheter's really for the benefit of the clinician more than the embryologist. I have to say that with some catheters, we had to modify our use of them as an embryologist because, for example, if you're loading as we used to, if you're loading from microtrops under oil, if you have a very flimsy catheter without any structural support, the medium oil interface just deflects the catheter. So it's very difficult. So we used to just take the cover of the inner catheter, cut it down at the end, and use that as external support for the inner catheter to load the embryo. So apart from that difference between supported and unsupported soft catheters, there's really no difference for the embryologist. It's really for the benefit of the clinician, and that's why you'll find that even in the same center, you'll find different clinicians having a preference for different types of catheters.
So with regards to sometimes we do face hatched blastocysts and we have to load them. So your practical tip in loading hatched blastocysts for the embryologists and also for the clinicians, because sometimes we don't know, are we causing more harm by more pressure while injecting, while doing a hatched blastocyst? So can you suggest some important tips?
Yeah, very good question. I think for the embryologist, I mean, hope that all embryologists are gentle when they come to load the embryos, and I'm sure they are. But obviously, if the embryo or the blastocyst is hatched from the zona, then they just need to be a bit more careful in the loading. And I think you're probably right. I think that the injection speed becomes even more relevant. I'm just guessing here, but it's just logical that the injection speed would become more important with a hatched blastocyst versus one that's protected in part or fully by the zona. So I think there you have to be really gentle in terms of injecting the embryo into the uterus. You don't want to bash it around on its way in there.
Thank you. So someone has asked adenomyotic uterus, when there is increased uterocervical length, any specifications in technique? I think you need to have mock embryo transfer. Having measured that, the cavity is larger, then you probably, can have
you'll know.
which are appropriate.
Frankly, you will know more about this than me. But again, I've worked with clinicians where they do a mock embryo transfer for all their patients when they're, coming through for their initial investigations. Part of their initial investigation is to do a mock embryo transfer, so there are no nasty surprises when it comes to actually doing the embryo transfer. And of course, you've got this all important embryo that you don't want to mess around too much when you come to do the embryo transfer. So again, that's a very relevant question. And again, you need to decide whether you're happy with going with the added expense of doing your mock transfer. But I can see the benefit in doing that.
Just can you tell us about the current scenario of embryo glue? Because there are a few people who are saying that embryo glue, without it, also they're getting good results. So I think there's a lot of evidence now for embryo glue in certain subsets of patients. So just give us some insights on using embryo glue.
Okay. Well, I'll give you some background. Well, I'll give you some examples, and then I'll give you the scientific background from my perspective. Just briefly, like I said, I have actually written an article on this. But in terms of the evidence actually does support the use of embryo glue. But I think you're right that it's more. The evidence is stronger in subgroups of patients that wouldn't do so well in a normal scenario. What's very interesting is I'm not sure if there have been many published studies on this, but what's very interesting is that embryo glue and uterine transfer medium, for example, they differ quite widely in terms of both the concentration of hyaluronan and also, if you like, the molecular weight of that hyaluronan, and also, one's in its salt form, which dissolves obviously quite easily in media and in uterine fluid. So, in those studies where it's been presented comparing the two, interestingly, in quite a number of them, no difference has been found between the two. So they're equally effective, if they're effective at all. And in terms of the scientific basis behind all this, I think it's a bit unfortunate that it's called embryo glue, because that's not where I think, based on my understanding and what I've read, that it's most likely to be beneficial. I think that it probably is beneficial to implantation, but not in attachment to the epithelium of the uterus. I think that the most of the evidence suggests that it's actually the stromal cells of the endometrium that secrete the majority of hyaluronan. And if you think about the syncytial trophoblast that needs to invade the stromal cells of the uterus, something like hyaluronan can serve a really good purpose. We know that hyaluronan is used in very many other ways to increase the space in tissues, and I suspect that that's what it's doing in the endometrium, that it's increasing that space between the stromal cells, making it easier for the syncytial trophoblast to invade the stroma or the decidua of the endometrium. So I'm not discounting it, but I think it actually serves a different job to what we maybe a lot of us believe it does. So just something to bear in mind. So I think it's it's a healthy approach to think it through and think where it may be most useful, and maybe to just, in terms of cost benefit, to use it where it's more likely to be benefit.
Thank you so much, Dr. Stephen. It's 45 minutes past midnight for you, and your thoughts and
Yeah. That's
tenure.
fine. I've
It
enjoyed
is
every minute.
overflowing. And sorry that we've been bombarding you with questions. We really took home a lot of scientific insights from your lecture today. On behalf of entire organizing team, myself and Dr. Sindhuja take great pride in thanking you for accepting our invitation and for doing this masterclass. Wishing you a wonderful Christmas and a very, very happy New Year 2026. Wishing all the delegates as well. So we will soon catch up in the next masterclass. Thank you so much, Dr. Stephen.
Thank you very much, Vasundara, and thank you everyone, and really good attendance turning up to listen to me. It's sparing your time, and yes, I just hope it was useful in one way or another.
I wish you a good morning or good night. I don't know.
Yes, it's good morning now, but like I said, it's not too bad. Don't worry about it, and hopefully see you in India or in London at ESHRA. One or more of you
Yes,
would be
yes.
great. Come and
Yeah.
say hello. Yeah.
Thank you so much.
Okay.
Thank you, sir.
Welcome. Thank you very much.
Thank you.
Bye-bye then.
Bye.
Bye.